Our next speaker, our third speaker in this first session is Irene Papatheodorou. So Irene is currently the gene expression team lead at EMBL-EBI. And I saw on Twitter that you'll move very soon, right, to the Earlham Institute, where you'll be the head of data science there. So today we'll hear about methods and tools for quality control and processing of single-cell data. So thank you so much.
Yeah, thank you very much for the invitation. I'm really happy to present here. First of all, I'm going to talk about moving on the analysis from raw data to count matrices. And in parallel, I'm going to give an introduction to the standardized analysis pipelines in single-cell expression atlas. So just to begin with, just to say where we come from and what we want to do with the team at EBI is our mission is to provide to the scientific community freely available information on the abundance and localization of protein and RNA across different species and biological conditions. And fair data is very important to us because then bringing it together, we can enable our users to answer questions such as where is my favorite gene expressed and how its expression changes, for example, in a disease. So in Expression Atlas and the Single Cell Expression Atlas especially, we collect lots of data from all kinds of species, and we also include a few available single cell data from plant species as well. And at the moment, you can find data sets from 21 different species and around 10.5 million cells. How do we get there? So how do we collect this data and what do we do to them? So what is our data flow and the journey? So we collect the necessary metadata for reanalysis, and having good metadata, as the previous speaker mentions, is hugely important for our purpose. And then we develop these standardized analysis pipelines, which I'm going to talk about later, soon, where we reanalyze all the data sets in a standardized way. And we develop the necessary web interfaces and search functions to enable these searches across the data. So enough about the atlas. I want to talk more generally about the single-cell RNA-seq analysis. And I'll begin by talking about what is different for single-cell versus bulk. And then what are the steps on the raw read processing, some basic QC, normalization, feature selection. I'm going to end up with some common file formats and tools for the matrices and the other metadata that are associated. So what is different in the analysis between bulk and single-cell RNA-seq? So basically with bulk RNA-seq, you have a lump of cells, which is a tissue section, and it all is mashed up, and you prepare a cDNA library that has basically cDNA from different cells within that tissue sample. And then you do the sequencing and you get different reads. And then basically for the analysis, there are two different choices. You can align the reads to a genome and then see which genes they come from, and then count them and quantify the expression level based on how many reads align in a specific gene, and get this expression matrix, which is samples versus genes, gene expression. Or instead of aligning to a genome, you can perform alignment-free transcript quantification and end up again with an expression matrix that will show you transcripts instead of genes. Now, as you probably know, there are different types of single-cell RNA-seq. And first, I want to talk about the analysis strategy of the SMART type single-cell RNA-seq methods because that's quite actually similar to the bulk RNA-seq analysis. So here you have this extra step. You start from your tissue sample, right? But you have a disaggregation step where each cell basically falls into a well. And then the libraries, you get different libraries, one library per well. So libraries are contain fragments by well and reads, and then you sequence. So you get a collection of FASTQ files that each one corresponds to a different well, so which is a different cell. And with smart type single cell RNA-seq, you get full length reads like in bulk RNA-seq. So really, once you have these FASTQ files, the rest of the analysis is quite similar to bulk RNA-seq. You have two choices again. You can align on the genome and get the expression matrix, or you can, which is going to be basically each sample is going to be a cell, right? Or you can perform transcript quantification and also get an expression matrix of the transcript. So on the other hand, in single cell RNA-seq, we also have droplet-based techniques. So with the advancements of microfluidics, we can basically have distinct barcoded beads where the cells get attached, and then these form droplets that then can be sequenced. And then that kind of so basically we start again with our tissue. We have this disaggregation and encapsulation into these oil droplets, and then here we have one library with all the fragments, which are reads by droplet. So what's happening here? We have barcoded the cells with these beads, with the barcodes on these beads. So that's how we can then distinguish by handling the barcodes. We can distinguish the reads by each cell, and then reach basically the point of the analysis where we were with the smart-like protocol. And then the analysis, the rest of the analysis is again alignment or transcript quantification and counting. So one of the pros of the droplet single cell RNA-seq is that it's applicable to a wider range of sample types than the smart-like. And also it produces a higher throughput, so a lot more cells because of this encapsulation and so on, and also the fact that you have one cDNA library at the end. The limitation is that here, unlike with the smart-like protocols, you don't sequence the whole length of the transcript, but you sequence only a single end, so either the 5' end or the 3' end, depending on the type of protocol. So basically, you cannot really identify well the alternative transcripts and so on. But you can identify more cells and potentially rarer cell types. So the length of barcodes, as I said, with 10x and droplet-based, we have these cell barcodes that we use to assign reads to cells. And also we have these UMIs, the unique molecular identifiers that basically tag cDNAs before amplification. And the value of this is that they give us an idea later on when we analyze the data for any amplification bias, so we can estimate that easier and then account for it. So depending on the technology, the barcodes will be in different files that also need to be handled by the analysis tools, so that then you can demultiplex basically the big file of the library into the different reads, and yeah, so then I think we can move now to the raw read processing. And so what happens, what are the tools basically that we use for alignment and counting when we have our reads and we have demultiplexed and everything. So basically here there is a variety of analysis tools, and they all have similar really common features. They handle the cell barcodes, and some may include correction, as I said, using the UMIs or not, so it depends on the tool. They perform some kind of transcriptome or genome mapping, again, depending on the tool. UMI deduplication, and they also perform counting. And what are the differences? So the differences really are in the order of the processes, like what they do first of those few processes. And also the main difference is, what we call monolithic tools, or tools that include separated steps. So monolithic tools is basically one tool that does everything for you, whereas other tools perform one step or two steps, and then you need another tool to perform the counting, for example. So in this kind of schematic, I'm showing the different, the main options. There are others as well. So you have the 10x cell ranger, for example, that is what we call like a monolithic tool. And it starts with transcriptome and genome mapping, barcode and UMI filtering and correction, then UMI deduplication and cell filtering, and then outputs the expression matrix. And then you have UMI tools that do the barcode extraction, counting, and UMI deduplication, Callisto and BUS tools that do pseudo alignments. So these ones don't use a genome to align. And they then perform barcode correction, they sort the cell, and then they count the reads. And then StarSolo is another tool that pretty much starts with the barcode extraction and correction, and then performs a genome mapping. So I think you get the idea. And then we have Alevin, which is actually the one we use in single cell expression atlas for 10x and droplet datasets, whereas for the smart-like datasets we use Callisto. So Alevin also performs similar functions, and in particular what we're using is Alevin Fry, which is the latest version of Alevin. And this is an image basically from the paper that shows the workflow of the tool, where you input the raw FASTQ files and also the indexes, the genome indexes, so that it can perform the mapping and the rest of the processing, including the quantification. And at the end, what it outputs is cell by gene count matrix, where it can tag ambiguous reads, unspliced or spliced, and you can use this to do further downstream analysis depending on the data set that you have, whether it is single nucleus, whether it's single cell, and so on. So now in the last bit of the talk, I would like to touch a little bit on basic quality control ideas. So basically, what do we want? What do we want in our data? So we go with the assumption that each droplet, when we deal with 10x data, contains mRNAs from a single cell, and we really want to we want real single cells. So therefore, we have to do this cell barcode correction, detect any droplets that don't have cells, so empty droplets, for example, and then detect these cases where you have doublets. So instead of one cell per droplet, you could have like 2 cells. So we obviously want to be able to take these and remove them from the data set, because they can obscure the rest of the analysis. And also we want to be able to detect any low quality cells and remove them. And also on the other hand, we want to estimate real transcripts. So basically, we want to check the resolution, check the UMI's deduplication, so that we're sure that we're not actually detecting increase in expression because of the amplification that is done for the sequencing, and also remove any ambient mRNAs. So Alevin Fry does perform, and not only Alevin Fry, pretty much all the tools perform some of these corrections, and they can detect doublets and so on. And basically, I would urge you to have a look at the publications of these tools to find out how they work and their workshops. So Cell Ranger also produces this kind of barcode rank plots that can give you an idea of where you should put your threshold for on the UMI counts, for example, in order to get an idea of what are the real transcripts and what is caused by the amplification, and also gives you a lot of other criteria that you can use to QC the data with respect to mapping and sequencing and the different samples. So I would also like to mention empty drops that we're also using, which is a technique to distinguish cells from empty droplets in 10x data and other droplet-based data sets. And this is the publication, but there's also a tool associated with this. And yeah, basically just to go a little bit deeper on how we detect doublets. So as I said, a doublet is basically 2 cells within a droplet with 1 barcode read. And what the algorithm does is use the original data to simulate doublets, so creating this kind of artificial doublets from cells that we know are single, and then perform dimensionality reduction together with the newly analyzed data. And then you would see the doublets in the sample clustering together with the artificial doublets that you have generated in the data set. And then you can identify them and exclude them for further analysis. And I would also just to note that this website, the single-cell-best-practices.org, is a nice resource for anyone to have a look and go further into the different aspects of the analysis of single-cell data. So, when I was asked to do this talk, and I had to finish on matrices, so I didn't know if it was going to be on raw matrices or normalized matrices. So I thought I should have a couple of slides on the normalization, which is what you perform usually once you have your matrix, your first cell by gene matrix. So why do we need to normalize? So what is the challenge here? So basically, because we have various effects that can affect the levels of gene expression that may not just be biological, maybe because of the sampling effects or sequencing effect or technology effects and so on, technical effects. And that means we can have like a non-uniform variance because of these kind of effects. Sorry. But many statistical methods that will be used later on to cluster the data and do annotations and so on, assume that you have the same variance in the data. So what normalization does is really to try and correct for those effects by modifying the expression values accordingly. But really to retain the biologically relevant heterogeneity and remove these technical effects. And again, there are quite a few different methods for normalization. Others that you can perform on the matrix, like on the gene expression data and modify the matrix, or others that you can just perform on the UMI space, like Harmony, which is what we use in Single Cell Expression Atlas. And then just to talk a little bit about the purpose of feature selection, so basically feature selection aims to remove non-informative genes that might not convey any biologically relevant variation. And that is because we tend to detect a lot of different genes per cell type, and some of them vary and others don't, and some of them can be informative for annotating the cell types or studying what we want. And the idea of feature selection is really to identify these informative groups of genes, such as the highly variable genes that we can use to, for example, later on cluster our data sets. Finally, for the last couple of minutes, I would like to talk about the different file formats and tools that you can access these matrices, or in the metadata and the different raw data sets as well.
Irene, I'm sorry to interrupt you. We have maybe 30 seconds so that we have time to get to a few questions.
All right, maybe I will skip that. And what I would like to leave you really is with some further resources and some online courses at the EBI that might be relevant. So happy to answer questions.
Thank you so much. We have some great questions coming in the Q&A box here. So just to kind of bring a couple of them together, there are a number of people who are wondering how you might choose an analysis tool. So for example, if you were choosing between Aleph and Fry or Callisto and Bus tools, are you thinking about performance or accuracy? Or someone was even asking, does it depend on what species or what sample you generated your data from? What are your thoughts on how you choose?
Yeah, so that's a great question. Obviously, there are lots of benchmarks out there that you can use. What we did for our purpose, because we are like a multi-species resource, we wanted something that doesn't compromise on performance or accuracy. So accuracy comes first, but it needs to be good enough for most species. So we tested on a variety of different species. Performance plays a part, especially like how fast and the analysis happens, especially for us because we are doing a lot of data, but maybe for someone analyzing a limited type of data set, number of data set, that's not such a big issue. What we also like to see is that the tools are being maintained by the community well, because that means we can use them for a long time, right? And basically also what we would like to see is that, but that's again for our purpose, is we would rather have non-monolithic tools, kind of tools that can split the processes in steps so that we can have flexibility according to the species that we're using and so on.
Got it. Thank you. Our next question is thinking about normalization. So if you are combining multiple samples or replicates, do you normalize all the individual samples first and then you combine them, or do you combine them first and then normalize as a whole?
So usually what we do is check first if we can avoid normalization. So if we have two different data sets, for example, and we integrate them on the UMAP space, and we see that similar cell types align, if we can avoid normalization is good, because as I said, with it you might remove some biological heterogeneity. So I would say the way you do it depends on the data set. You do have to batch correct when you are doing like a meta-analysis, bringing together data from different studies.
Thank you. I think we have time for maybe one or two more quick questions. So the first one maybe is a quick question. We were talking about doublets. So how often does a doublet occur? And is that something that people should be concerned about in their data set, or is it a pretty rare occurrence? So for example, with 10x.
Yeah, so okay, so I cannot give you a number because I'm not we see them in most data sets, so that's why we have as routine the doublet detection and removal algorithm in the pipeline. So I cannot tell. It varies how many you get per data set, because I think it varies on the tissue as well. So that's why I cannot give you a figure, but it is fairly common.
Got it. Thank you so much, Irene. That's all the time that we have. But if you have time and you would like to go through and type answers to the questions that are in the Q&A box, we would really appreciate it. Thank you so much.
Thank you very much.